See here, nucleic acid. The term nucleic acid has been given by Frederick Miescher, and nucleic acid consists of nucleotides, we know, and nucleotides NT and NC. NC is smaller than NT, so you can easily remember this is the greater than sign. NC nucleoside is smaller than nucleotide. So tide means another part is there. Tide means nucleoside plus phosphate group. This is the phosphate group, addition of phosphate group, it will make the nucleotide. So nucleoside consisting of sugar, one sugar molecules, that may be ribose or deoxyribose, plus nitrogenous bases are present. Nitrogenous bases means any type of nitrogenous base, A, T, G, C, any combination of adenine, guanine, cytosine or thymine, any types of nitrogenous bases are present in case of nucleosides, anyone. So a number of nucleosides are attached to each other to make polynucleotides structures. Two types of nucleosides, deoxyribonucleosides and ribose. What is the difference? Deoxyribonucleosides, sugar is basically 2-deoxy-D-ribose. At the position of carbon number 2, the oxygen is deoxygenated, means oxygen is removed. It is double stranded. Another features, nitrogenous bases is purine, adenine, guanine, and pyrimidine is the cytosine and thymine. So DNA plus protein, basically histone protein, together form the nucleoprotein called nucleosome. This is a very good examples and questions. Polynucleotide. How polynucleotide? One by one nucleotides are attached to each other to make this polynucleotide chains. So it is found all over our body systems in the chromosomes. Ribonucleic acid, RNA. What is the difference between RNA and DNA? So many questions are coming nowadays. Commonly found in the cell cytoplasms. It is a single-stranded. That was a double-stranded. It is single-stranded. Nitrogenous bases is basically adenine, guanine, cytosine. These are the same compositions, but uracil is replaced by thymine. Sugar is the D-ribose category. The synthesis of RNA is dependent on DNA by transcription process. Through the transcription process, DNA can be converted into RNA. Bacteriophage, which have the single-stranded DNA virus. That is single. This is the example of exceptions. Vitamins. Vitamin was first introduced by Funk. The name. It is water soluble. It may be water soluble. It may be fat soluble. Water soluble vitamins are vitamin B complex. All the types of B vitamins are water soluble. B, C, and niacin. Niacin means D. Vitamin D. Fat soluble is A, D, E, K. You can be easily remember it. Vitamin A, D, E, K is the fat soluble, where the BCP is the water soluble. Vitamins B12 is only found in case of microorganisms. This is a very important. Vitamin B12, thiocobalamin, I think I have to see. I have to check thiocobalamin maybe. They are not found in higher order of the organisms. Only microorganisms have this particular vitamin B12 categories. Vitamin K1 found in the plants and vitamin K2 found in. This is the difference between K1 and K2. Vitamin K is fertility factor. K1 found in animal plants, but K2 found in animals. This is a slight difference in the structure. So these are the very important questions. What are the deficiency symptoms come due to a specific vitamins? A, xerophthalmia, nyctalpoia, and hemeroploia, and B1, thiamine beriberi, edema, B2, pellagra, alpha-thiamine acid, and B12, Pemiocin, anemia, B6, gastritis, niacin, dermatitis, vitamin B6, scurvy, very important, vitamin D, calciferol, ricket and osteomalacia. This is for baby, this is for baby child conditions, and this is for mature humans. Vitamin E, tocopherol, this is the fertility factors. Under the deficiency, sterility can become, hair fall can be possible, degeneration of muscle can be. So vitamin B, vitamin E is very important for our daily as a supplements. Vitamin K, phyloquinone, blood clotting factors, it is known as. Vitamin K is present, should be present, and vitamin K deficiency leads to continuous release of the bloods if some cuts will be happen. Enzymes, these are the special classes of proteins called biological middleman because they are conducting one experiments, one particular events that is called enzymatic reactions where they will act on the substrate plus enzyme. We can see it will finally change the conformation of the substrate to make this product. So a number of chain reactions are there. Enzymatic activity was first discovered by Buckner. This is also important. Zymase was the first enzyme found initially in the yeast. This is also another important point. The term enzyme was coined by W. Kuhnhe while working on the fermentation experiments. So these are the basic things as per their composition. What are the difference between holoenzyme, apoenzyme? We can see very from the beginning. So basically enzymes is a simple enzymes. We can say enzymes are proteinaceous substance. If enzyme itself is working without any type of units are attached to each other, this is the simple enzymes we can say. If enzymes are attached to another non-enzyme part, this is called this enzymatic part is called apoenzyme. Non-enzymatic part is called coenzyme. Okay, again the protein part, just say here enzyme is the protein part is basically called apoenzyme, and if it is replaced by the coenzyme is replaced by a particular group of proteins that is non-detachable, to this particular enzyme unit, then it is called prosthetic group. It is called, you can see here, prosthetic groups. The non-protein part that is not separated, that cannot be separated from the enzyme unit, then it is called prosthetic group. Coenzyme, a non-protein component of the enzyme, initially I told, which is an organic compounds. If it is organic, then it is called coenzymes. If it is inorganic or metallic ion, inorganic, IO, we can see this is called cofactors. That refers to the prosthetic group which is an inorganic ion. You can see here cofactors, COF, and this is COE. So these are the different, very easy differences. You can easily differentiate it among them. Ascorbic acid, oxidase, and catalase are the examples of cofactors. Some of the good examples and points are the enzymes can be associated with pure states, and it can reacts with both acidic and alkaline substances, show sensitivity to heat. You know the Michaelis-Menten equations, it can be easily come into your final exams. What are the significance of each and every units, K, M, everything. Enzyme activity can be accelerated by activators such as manganese. These are the metalloenzymes part, which is responsible for the activity of the enzymes in some cases. Enzymes are organic catalysts. This is the very basic features. So classification, this is the classification based on the IUPAC nomenclature. First classification you can easily remember by this very simple formula, OTHLIL. O means oxidoreductase, T means transferase, H means hydrolase, L means lyase, I means isomerase, and L means ligase. So what are the particular catalyzing reactions are given here, and then very good examples. You just have to remember, okay. And special classes of enzymes are isoenzymes, proposed by Marquette and Moller. They are the different enzymes which catalyze the same reaction in different tissues. That means the reaction can be occurred in different types of tissues, in such cases the enzymes will be the same. Then it will called isoenzymes. They are different in molecular weights and exercised from different genes. So that means enzymes are the different, but their functions are same. Okay, so enzymes are different. Enzyme 1 and enzyme 2 work on two different tissues or organs, and their functions are same. Allosteric enzymes, these are the regulatory enzymes which have more than one polypeptides. If it has more than one polypeptides and as well as allosteric category, allosteric means feedback inhibition it should have. This type of enzymes have well developed regulatory mechanisms. Okay, so this is called regulatory enzymes also. They produce a sigmoidal curve. Sigmoidal curve is like this. If it is y-axis, this is x-axis, then it will be like this. It will be increasing, then plateau, downward plateaus. Ribozyme, they are the catalytic RNA which have the enzymatic properties. This is the non-protein types of enzymes, only one that has a catalytic property activity, enzymatic activity. Factors affecting temperature, if the temperature increase, the catalytic activity of the enzymes is decreased after a certain time points. Many enzymes stop activity after 60 degrees centigrade and denatured. And second one is metals, showing variation in the activity when they attached with some different types of metals. Heavy metals, phytogelatin synthase, this is the enzymes. If it is interacting with a particular enzymes, it will chelate down the particular metals. pH factor, pH effects, if it has a specific pH, it will work. That's why it is also known as isolytic point. This particular each enzyme has a specific pH levels to work functionally good. Substrate concentration, it is also again important. If the substrate concentration is more enzyme is required, otherwise enzymes will be deactivated very fast. Enzyme concentration is also again, if the enzyme concentration is more, the chances of enzyme activity will be more. Effect of water activity, other points you can see here. This you have to be just remember, so many questions are coming in the last 10-year questions. I have collected these informations. All the things are informative only. You have to just remember one by one. Each and every questions are important. Who will be the father of modern enzymatology, with the father of biochemistry, chemistry. So today I have finished this particular lectures by 51 minutes, 51 to 52 minutes. This is the longest lectures I think, because a number of topics are there. So all the best for your next lecture videos, and try to recapitulate each and every points and ask me if you have any doubts. Thank you.